Thank you very much, Professor. It's my honor to present in front of our distinguished speaker and guest here in Doha, Qatar. Basically, I'm going to speak about our preliminary result of genetics of Brugada syndrome in the Gulf. We just started basically two months back, so we have very little information, but I would like to share with all of you with our findings. Basically, I will start where Professor Brugada ended, epilepsy, Brugada, and sudden death. In fact, this is a case from our hospital, reported at the American Journal of Emergency Medicine. A 30-year-old Egyptian young guy, he's a laborer, previously healthy. He came to us with almost 60 episodes of syncope. And we admitted him to the hospital, and we were talking among our group that it will be impossible for this guy to have the cause of syncope is due to ventricular fibrillation, and he is still alive. So we ran a battery of investigation trying to understand what is the etiology of his syncope. During tilt table testing, he started to seize, and he was observed to have a focal seizure. We looked at the monitor. He has no arrhythmias. He was hemodynamically stable. We had to stop the test. In minutes, he recovered back to his normal status. He was seen by our neurology colleagues, and they did an EEG, and they discovered that he has one form of epilepsy. His baseline ECG, he is manifesting type 1 Brugada ECG. So we discussed and reviewed the literature thoroughly regarding treating this patient. He had almost 60 episodes over 1 year. So we decided to go with this medication, oxazepine. After his second dose, he went into ventricular fibrillation storm. He was almost cardioverted 16 times. He kept going into VF storm until they called one of the electrophysiologists on call. We had to give him intravenous isoprenaline, where his arrhythmia been settled, and so we decided that we will implant a defibrillator. We gave him a full list of medication that he should avoid in the future. So basically, this is just a start. We are seeing quite frequent of this kind of presentation. So this has enlightened us to talking to my colleagues in the Gulf, in the region. We decided that we should study those group of patients. Sorry, this is not. Yeah. Okay, so discussing the idea with the QCRC, Professor Magdi Yaqub, and the electrophysiologist in the Gulf region to look at all patients with manifested type 1 Brugada syndrome, to include them in a genetic study. We are hoping to recruit 150 patients over the next 3 years. So our idea basically to identify the genetic cause in our set of patients. It is very unique in this region. As my previous speakers, we have patients from all over the world. We have a ethnicity difference from the Arab as well as from the Far East. So to know more about the disease mechanism, underlying differences, and to study the presentation and the pathophysiology. We have, as I indicated, we started 2 months back. We recruited 33 patients, but because, as you know, the time of processing takes time, only we had 11 results are so far. Out of these 33 patients, 10 are from Abu Dhabi and 23 from Doha. What we are analyzing is DNA. We extract whole blood and DNA analysis. So 11 out of 33 patients was sequenced using targeted next generation sequencing panel. And the panel covers the exome of 170 genes. And basically we are testing the core as well as the other reported genes, the SCN5A, as well as we are looking at the modifying genes. Okay, here is our work flow. So patient consent and pre-genetic testing and counseling attempted. So we take a peripheral blood as well as we enter clinical data entry. We take DNA extraction, next generation sequencing, and then we look at the bioinformatical analysis. And with the help of our QCRC colleagues, Khulud Al-Shafi'i, she is helping us in this aspect. So we divide them into 3 groups. A novel variant predicted to be pathogenic. And here we try to screen the relative or unaffected individual in the families. Whereas the other 2 groups, which are known variant, but they may be associated with other diseases, for example, HCM, as we will see in my next slide, or the 3rd group, there are known variant associated with Brugada syndrome. So we do a variant confirmatory testing. And following that, we do post-genetic testing counseling for patients and relatives. So this is our result. Out of our 11 patients, 4 of them tested negative, and 7 of them tested positive. I could go through the findings here. So this is our first patient. He is 29-year-old male Syrian with a spontaneous type Brugada on electrogram. He had a history of syncope, and he had a defibrillator implanted in 2008 and no ICD discharge since that time, and he has normal structure heart on echo. He tested positive for SCN5A, and probably he has a novel pathogenic variant. He's by himself in the country. His whole family in Syria. We were trying to get hold of his family member to screen them. But at this stage, it is not possible. The next case, he is another young guy. He's a 27-year-old male patient from Bahrain with type 1 Brugada. He luckily had a VF arrest while in hospital, and he had a defibrillator implanted in 2011. And following discharge, he had number of ventricular fibrillation and discharge from his device, which was appropriate, and we had to put him on quinidine. And tomorrow there will be more session discussing into the treatment therapies of Brugada. So this gentleman also tested positive HCN4, which is one of the gene modifiers, and he is possibly pathogenic Brugada, but it has been reported. So here we need to confirm the genetic variance in this patient. Patient number 3, he's a 32-year-old Indian male with a history of non-sustained VT. His baseline ECG showed the type 3, which was provoked by IV flecainide to type 1. He otherwise has a normal structure heart. This patient tested positive SCNB1 with a novel mutation, which is possibly pathogenic to Brugada. And what we are interested in this, we would like to functional validation of the variant effect. And we are going to use the zebrafish model, which is we are doing it for the first time here in at the QCRC and in Qatar. So we are. This is still pending. Patient Brugada 4. He is a 45-year-old. He's the only Qatari in our patient list here, with a history of palpitation and ECG of type 1 Brugada with a normal structure heart. He reported to have MYBPC3, which is probably reported pathogenic cardiac, and we needed to check for hypertrophic cardiomyopathy. Is there a relation between Brugada and the hypertrophic cardiomyopathy? We are going to do a cardiac MRI, and but based on echo, there is no evidence of hypertrophic cardiomyopathy. The next patient, Brugada 7, he's a 34-year-old Indian male with a provoked type 1 Brugada ECG with positive family history of sudden death and history of VF arrest. He was a successful survivor, so he underwent an ICD implantation recently in 2000 this year. He is also tested positive for SCN5A, and there we need to confirm him with the zebrafish for functional validation of variant mutation. The last two patients here, we have a 51-year-old Pakistani male patient with a history of VT and type 1 Brugada on ECG, otherwise normal structure heart. He tested positive with FOXD4, and he confirmed the genetic variant. And our last patient is a 35-year-old Malaysian with a history of palpitation and type 1 provoked and normally structure heart. He tested positive with MYBPC3, and we need to again this is pathognomonic for HCM. We are investigating him for HCM as well. Looking at these patients here, we have a Syrian, Bahraini, Qatari, Indian, Malaysian. So there is a quite ethnicity in our patient population. So regarding the zebra model, so the credit here goes, this slide is a courtesy of Sahar Isa. Basically, what we, in patient 3 and 7, we take the blood sample for DNA sequencing, and we send them for genomic engineering. This is the fish embryo with a DNA what you call this, Sahar, can you? Yes, the morpholino, and we get we have the result here of elimination of gene function or report gene function or alter the gene function. So based on these findings, this is what fascinates me as a clinician as well as scientist, where we sit together and we can plan therapy for patients by incubating the petri dish using medication with the embryo of the zebra fish. And basically what we are looking at the potent phenotype mutation that these drugs are able to eliminate those phenotype mutation back to normal function. From this, we could engineer medication that will even help in the treatment of these patients. So this is just why zebrafish model has been used using genetic. This is using the wild type with the stripes here. So we get elimination of the melanin and stripes here, and you could see here the Casper mutant. It becomes transparent, and we are able to study this quite clearly. So this is again the same principle of the morpholinos and the DNA. Here is the mRNAs with the morpholinos are attached at the start site or spliced site, injected into the embryo, and there is the morpholinos with the mRNA where it impairs the protein synthesis. This is just a movie here where it shows the altered heart function in a zebrafish model. This is in a normal heart, and this is in a diseased heart fish. You could see it is dilated here, and whereas here it is normal in size. So our conclusion, the Gulf Brugada genetics, although small in numbers, but novel genes have been identified, and there's still much more to learn about. There is expanding genetics, and the role of ASEAN-5A is unclear. There is much more to genes are playing role in this part. Symptomatic patients are at risk of sudden death, and they need an ICD. This will be also talked about tomorrow. And the asymptomatic patient, the risks are still ill-defined, because they may be asymptomatic now, but in 1 week or 1 month they may be presenting their first symptom as a sudden death. Thank you very much.
Yeah, first of all, to congratulate you and your team for being doing all these jobs. I think it's important that we understand. Yeah. As we were saying, I think we are repeating this all the time during the session today, that we need to know, we need information, and of course the research is the basis of this information. So thank you for this information. My question goes to the 2 patients that had a myosin mutation. Is this because how do we relate that to the Brugada? You say that it has to be excluded hypertrophic cardiomyopathy in these patients. Clinically, you say that they do not have structural heart disease. You need a nuclear magnetic resonance in these patients to exclude some fibrosis and some
Not yet. Yeah, as I said, this is just, we started 2 months back, and it is just right at the beginning, but as part of the workout, we are planning to do cardiac MRI for those patients.
These patients who have some electrocardiographic signs or possible hypertrophic cardiomyopathy or not? It's not unusual that these hypertrophic cardiomyopathy patients that have an ECG which is abnormal, but they do not have the phenotype on the echocardiogram, but then you go to the molecular magnetic resonance, and then you start doubting because you see a little bit of fibrosis in the setting.
Yes, we have a group, actually a bigger group in the HCM where they are, and that's well known, of course, genotype positive, phenotype negative. And there has been Caroline Ho, for example, from Harvard, has shown that actually, if you look for pro-fibrotic markers, in this group of patients, you have high pro-fibrotic markers, so there could be fibrosis. Certainly, these patients are going to undergo MRI and look for that. And the other important thing is with the 170 genes, we're looking for modified genes to see. But the interesting thing is that they do have ECG changes of Brugada. So that is the interesting thing. But we will.
All these overlap of syndromes that we are talking all the time, that we are trying now to understand. As we get more information, we get more trouble also to solve them.
Questions from the floor? You seem to have convinced them.
I just would like to acknowledge our team here, which I basically from the Heart Hospital, Dr. Jassim Al-Suwaidi, Shahul Hamid, Sayyid Abdurrahman Qasim, Muhammad Mohsen, Dr. Jassim from UAE, and I'm sure maybe the other Gulf state, Dr. Alawi and Dr. Salim Al-Ka'bi from QCRC. Special thank to Khulud, Sahar, Christian, and of course you, Professor.
One final question, short.
This is the trouble guy here.
The biophysics man.
Make it easy on me or otherwise.
The biophysics man.
I will try to make it as easy as possible. But it's just from the perspective of somebody who wants to do translational study, right, from the animal to humans. Well, if you, let's say you have protein C mutation that works very fine on the animal, which is zebrafish, knowing that zebrafish heart is only one ventricle, and the heartbeat of zebrafish is completely different from human. In addition, the temperature, working temperature of zebrafish is completely different from human. So how would you do the transition between what you found on the zebrafish back to the human who have two ventricles and two atriums?
Sahar is dying to answer you because there has been work in the literature on myosin binding protein C with morpholinos, and Sahar is already doing some work on certain of mutations which has been encountered in our patients. These are HCM patients, and she has preliminary results showing actually that these mutations can be reproduced with a morpholino. You reproduce the phenotype. What she's trying to do is to correct the phenotype with human myosin binding protein C. Am I right, Sahar?
Yes.
Can you explain yourself a bit more?
Of course. So just as zero pressure, as a situation, what we do is using this whole genetic manipulation to eliminate the gene function in the fish. We model the human mutation in the fish. So by injecting the morpholino, either we remove the function of the gene, which will be a complete knockout, or we alter the gene function. Once you have the embryo, which we manipulate as having the same symptoms as the patient, this embryo will be stable to test for drugs. So in a small 96-well plate with pre-approved, like FDA pre-approved drug libraries, we can screen for hundreds of small compounds or already pre-approved drugs. And this is the beauty because you do it for a person, individual patient. So now we are going into a personalized patient-specific therapy that will be just specific to that patient. What we do first, we align the human gene with the zebrafish gene, see how much they share in identity, and we try to rescue the expression. So with the myosin binding protein project, what we did is we cloned the human genes with the different mutations, and once you eliminate the function of that gene in particular in the fish, you inject the RNA of the human gene and see whether it will rescue the phenotype or not. Are we getting that abnormal heart back to normal or not? And you validate that with different molecular pathways. With the zebrafish as well, we can do microarrays, so we can test of different associated genes. Let's say we have a sodium channel mutation, but it is associated with another thing. We can get the answer by the microarray done in particular fish, modeling that particular mutation.
She answered your question beautifully. But there has been also in the literature, while Sahar and the group here were trying to reproduce HCM in a zebrafish, there has been a publication in circulation from China doing exactly that. They did a morpholino, myosin binding protein C mutation, no silencing, complete, Sahar, isn't that right? They silenced the whole of myosin binding protein C in the zebrafish, and they've shown problems with both relaxation, the heart was not functioning properly, and they reported as well arrhythmias in a myosin binding protein C mutation, which is not mutation, it is silencing in the morpholina in a zebrafish.
I want to mention just one last thing. For the long QT syndrome, there is already a zebrafish well-established in Newton, and they did drug library, and they did get another chemical. It's now in Harvard moved to clinical phase 1 to be tested in patients, and it's called fluor and renal light. I don't know if you did hear about it, but the zebrafish beauty is.
You just like zebrafish. Okay. With that, I think we thank the speakers, wonderful speakers, and the participants. Thank you very much indeed. And we see you tomorrow. Thank you.